in the high-altitude grasslands of the Andes around 6,000 years ago. A guanaco stopped running. It did not flee from the humans moving quietly through the puna grass. It did not charge them either. It stood its ground at the edge of a human camp and waited. Nobody knows why. Nobody knows which guanaco or which night or which camp. But that moment, whenever it happened, set something in motion that would outlast every empire that has ever risen and fallen in the mountains of South America. The descendants of that guanaco would carry loads across the highest roads on Earth. They would feed civilizations that left no written records. They would become the spiritual and economic backbone of the Inca and every culture that came before them, stretching back into a past so deep that the ice itself remembers it. We call them llamas, and for most of modern history, we thought we understood exactly what they were and where they came from. We were wrong about almost all of it. The genetics of these animals has spent the last 2 decades dismantling everything we assumed about the origins of South American camelids, where they came from, when they emerged, how many times domestication happened, even what the word llama genetically means. The picture that has emerged is stranger than any of the popular theories. It is also, in many ways, more disturbing, because the story of the llama is the story of an entire species, and most of that species is gone. The ghosts of vanished lineages are still hidden inside the DNA of every modern llama alive today. And only recently have scientists been able to read them. This is what the ancient DNA actually shows. Let's begin. For a long time, the question of where llamas came from was treated as largely settled. The story was simple, and it was told with confidence. Llamas were domesticated from the guanaco, a wild camelid that still roams the Patagonian steppes and the high Andes today. Alpacas, their smaller cousins, were domesticated from the vicuna, a delicate, slender, wild camelid that lives at even higher altitudes, grazing on the freezing, wind-scoured puna above the tree line. 2 wild ancestors, 2 domestic descendants. It was clean. It was symmetrical. It made sense. And for decades, nobody had any reason to doubt it. The physical resemblance between a guanaco and a llama is obvious to anyone who has seen both animals. They share the same upright posture, the same coarse fleece, the same long neck and large, wary eyes. Guanacos are wilder, leaner, and more agile, but the family resemblance is unmistakable. Likewise, the vicuna and the alpaca are clearly related. Both are smaller, finer-boned, and produce incredibly soft fleece that has been prized since the Inca and before. The vicuna's fiber is so fine that it was once reserved exclusively for Inca royalty, woven into garments so soft they were said to feel like woven cloud. It all fit together neatly. The domestication was assumed to have happened in the highlands of Peru or Bolivia, perhaps 5,000 or 6,000 years ago, as early Andean societies began to settle and manage animal populations. The problem was that almost everyone was looking at the wrong kind of data. For decades, the entire field relied on modern animal DNA, working backwards through the family tree to guess where the branches had started. The assumption was that the modern domestic animal must point clearly back to its wild ancestor. That assumption was wrong. Modern llama and alpaca populations have been shaped by thousands of years of mixing, replacement, and most recently a catastrophic near-extinction event that scrambled the genetic signal almost beyond recognition. Reading modern animals to find ancient ones is like trying to read the original draft of a book by looking at the 17th edition. The only way to find the real story was to stop reading modern llamas and start reading the bones of dead ones. That is exactly what a team of researchers set out to do. In 2020, a study published in the journal eLife by a team including researchers from the University of Chile and the University of York, working with ancient DNA extracted from archaeological sites across the Atacama Desert and the Andean highlands, set out to trace the true domestication history of South American camelids. The team sequenced mitochondrial DNA from ancient camelid remains, spanning thousands of years, and compared them to modern llamas, alpacas, guanacos, and vicuñas. They included samples from the Tulán ravine in northern Chile, one of the earliest known sites of camelid management, dating back roughly 3,000 to 4,000 years. It was the most comprehensive look at camelid ancestry ever attempted using ancient genetic material, and the results were not what anyone had predicted. The headline finding was immediately baffling. When the team compared the ancient DNA to modern animals, they discovered that the neat symmetrical story of two wild ancestors giving rise to two domestic descendants did not hold up. The ancient DNA showed something far more tangled, far older, and far stranger. The first finding overturned the single origin theory for llamas. For years, researchers had argued about whether llamas were domesticated once or multiple times. The prevailing view leaned toward a single domestication event, probably in the central Andes. The new study found the opposite. The ancient DNA pointed to two distinct guanaco lineages contributing to the domestic llama. Two separate wild populations, likely in two different geographic regions, had been domesticated independently and had then merged over thousands of years of interbreeding. Llamas were domesticated twice, not once. Two events, two populations, two beginnings that converged into the animal we know today. This finding alone was enough to restructure the entire field. But here is where the story becomes strange. The alpaca, the small, soft-fleeced camelid that every textile expert and every tourist in Peru recognizes, was not what anyone thought it was. The old consensus said alpacas came from vicuñas. The DNA said something else entirely. When the research team analyzed the genetic signatures of ancient and modern alpacas, they found that the vast majority of modern alpacas did not cluster with the vicuña. They clustered with the guanaco and the llama. Modern alpacas were, genetically speaking, not a separate domestication of the vicuña at all. They were a variant of the llama lineage, or more precisely, the product of extensive hybridization between llamas and vicuñas over thousands of years. The alpaca as we know it may not be a pure domestication of any single wild species. It is a hybrid creature, a blend of two wild ancestors that was then shaped by human selection for finer fleece. This is not a minor correction. This is a fundamental rewriting of what the alpaca is. For decades, breeders, textile experts, and scientists had organized the entire camelid family around the assumption that the alpaca was the vicuña's domestic descendant. The DNA says that is not true, at least not in the way anyone imagined. The alpaca is something stranger, a human-made blend that carries the DNA of two wild camelid species, a creature that does not map neatly onto any single wild ancestor. And here, the story becomes darker. Because the original alpaca lineage, the one that may have been a genuine domestication of the vicuña, is almost entirely gone. Read that again. The alpaca that the Inca bred, the one whose fleece was reserved for royalty, the one that was woven into the finest textiles of the pre-Columbian world, that animal's genetic identity has been almost completely erased. What happened to it is a story of conquest, catastrophe, and deliberate destruction. When the Spanish arrived in the Andes in the 1530s, they encountered the most sophisticated animal husbandry system in the Americas. The Inca and their predecessors had managed camelid herds for thousands of years. They had breeding programs that would not be matched in sophistication anywhere in the world for centuries. They maintained strict separation between llamas and alpacas, between wild and domestic populations. They tracked lineage, culled undesirable traits, and produced animals of extraordinary quality. The Spanish did not understand what they were looking at. They saw camelids as inferior to the sheep, cattle, and horses they had brought with them. They did not value the alpaca's fleece, which was finer than any wool Europe had ever produced, because they did not know how to process it. They wanted European wool for the textile trade, and they wanted to establish cattle and sheep on the high-altitude pastures that had sustained camelid herds for millennia. And so, as a matter of deliberate policy, the Spanish colonial authorities dismantled the Inca breeding system. Herds were confiscated, dispersed, and slaughtered. Camelid populations were deliberately reduced, both to make room for European livestock and as a measure of social control, to destabilize the indigenous communities that depended on them. A study published in 2018 in the journal Genes by an international team including researchers from the University of Chile documented the catastrophic genetic consequences of this collapse. Within a few centuries of the Spanish conquest, the carefully maintained genetic boundaries between llamas and alpacas broke down completely. Animals that had been kept separate for thousands of years were forced together in shrinking remnants of their former herds. Hybridization, which the Inca had rigorously prevented, became universal. The original alpaca lineage, the one that carried the genetic signature of a true vicuna domestication, was swamped, diluted, absorbed into the larger llama gene pool until almost nothing recognizable remained. The modern alpaca is, in most cases, a hybrid animal that carries predominantly llama DNA with varying degrees of vicuna ancestry. The fine fleece that distinguishes it from the llama is the product of human selection applied over centuries to a mixed origin population, not the inheritance of a pure vicuna ancestor. The original alpaca, the Inca alpaca, is a genetic ghost. Its DNA survives only in fragments, scattered across modern herds, mixed beyond recognition into an animal that looks right but is not what it was. This is not extinction in the usual sense, it is erasure. The animal still exists, but its genetic identity has been overwritten by another species' DNA, the result of a colonial catastrophe that destroyed not just a civilization, but an entire branch of domestication. The alpaca standing in a field in Peru today is a survivor of that catastrophe, but it is also a testament to what was lost. There is yet another layer to this story, and it is one that researchers are still struggling to fully understand. When the research team examined the genetic relationship between wild and domestic camelids over time, they expected to see what every other domestication study has shown, that domestic animals exchange genes with their wild relatives over time, picking up wild DNA whenever domestic populations encountered wild ones. They saw something different. The ancient DNA from the Tulán sites showed that early domestic camelids carried genetic signatures that were distinct from the wild guanacos living in the same regions. The domestic and wild populations were genetically separated, even when they lived in close proximity. This suggests that the early Andean herders were actively managing their animals' breeding, keeping domestic and wild populations apart in a way that was virtually unprecedented for the time. The Inca inherited and perfected this system thousands of years later, maintaining strict breeding records and enforcing separation between llamas, alpacas, guanacos, and vicunas. But the genetic evidence from Tulán suggests that this practice was far older than the Inca, far older than any Andean civilization we have a name for. The people of the Tulán ravine, living in the Atacama desert 3,000 to 4,000 years ago, were already practicing a form of controlled breeding that kept domestic and wild camelids genetically distinct. This is one of the earliest examples of deliberate genetic management by humans anywhere in the world, and it may explain why the domestic llama and alpaca were able to maintain distinct identities for thousands of years before the Spanish conquest shattered the system. The DNA cannot tell us exactly how they did it. It can only tell us that the separation was real and that it held for longer than most human civilizations have existed. The site of Tulán itself adds another dimension to this story. Located in the Puripicar volcanic field, the ravine was a harsh, arid environment where water was scarce and survival demanded intimate knowledge of the landscape. The archaeological deposits there revealed dense layers of camelid bone, ash, and fiber, evidence of a community whose entire economy revolved around these animals. Researchers found corrals built from stone, their walls still standing after 4 millennia, designed to contain and separate herds. They found processing tools made from obsidian, with edges still sharp, used to shear fleece and butcher meat. The floors of these corrals were packed with dried dung, preserved by the extreme dryness of the Atacama, so perfectly that scientists could analyze its botanical contents to reconstruct what the animals ate. The diet was varied, including grasses from the high puna and wetland plants from the ravine floor, evidence that herders were moving their animals across different ecological zones to maximize grazing. This was not opportunistic hunting. This was landscape-scale management, a system of rotational grazing and controlled breeding that predates the earliest known pastoral systems in Europe by centuries. The people of Tulán were not just keeping animals, they were engineering an ecosystem. The ancient DNA also revealed something unexpected about the geographic spread of these early domestic lineages. When researchers compared camelid remains from Tulán to samples from sites farther north, in Peru and Bolivia, they found genetic connections spanning hundreds of kilometers of the most extreme terrain on Earth. Domestic camelids from the Atacama were sharing genetic signatures with animals from the highland Puna near Lake Titicaca, a corridor of exchange that linked desert communities to highland pastoralists through routes that climbed past 4,000 meters in elevation. These were not casual trading relationships. The genetic evidence points to a structured network in which breeding animals were moved across vast distances, likely along the same natural corridors that would later become the Inca road system. The communities of the pre-ceramic Andes were already moving animals along the routes that the Inca would pave with stone thousands of years later. Further south in the Patagonian lowlands, a different story was unfolding. Guanaco populations there were never domesticated, but they were managed in ways that left detectable traces in the archaeological record. Hunting parties used corrals and drive lines, stone structures built to funnel wild guanacos into controlled spaces where they could be selectively culled. The bone deposits at sites like Cueva Fell, dating back over 10,000 years, show that early Patagonian hunters were targeting specific age and sex classes of guanaco, a form of population management that stops short of domestication, but reveals a deep understanding of animal behavior. The guanaco was not simply hunted, it was monitored, managed, and in some cases protected from overexploitation by people who had no word for conservation, but practiced it nonetheless. For most of their history, llamas and alpacas were bred for function. Llamas were pack animals carrying goods across the Inca road system, the most extensive network of high-altitude trails in the ancient world. They were bred for strength, endurance, and sure-footedness on terrain that would kill a horse. Alpacas were bred for fleece, for the extraordinary fineness of their fiber, which was measured in microns and woven into textiles of a quality that has never been matched. Then, within the past century, almost everything changed. When alpacas were exported from Chile and Peru to the United States and Europe in the 1980s and the 1990s, a new market was born, driven not by function, but by aesthetics and investment. In North America, alpacas were marketed as exotic livestock, promising high returns through breeding and fleece sales. Llamas underwent a similar transformation, becoming show animals and pets rather than working pack animals. New breed registries were created, strict standards were established for fleece density, crimp, and conformation. Animals were selected for traits that had nothing to do with survival in the Andes, and everything to do with what appealed to buyers in a completely different hemisphere. The North American alpaca population was founded from a small number of animals exported from South America, creating a genetic bottleneck that reduced diversity. Selective breeding for specific fleece traits further narrowed the gene pool. Today, many alpaca herds in North America and Europe carry significant burdens of inbreeding, with effective population sizes far smaller than those of their ancestral Andean counterparts. When you look at a show alpaca in the United States, you are looking at an animal shaped by a market that did not exist 50 years ago, bred for a fleece standard defined by a registry that did not exist 40 years ago. Its genes were shaped by the high Andes, but its body has been remodeled by an industry that has no connection to the ancient herders who created the animal in the first place. The broader camelid family has its own deep history, and it is one that puts the Andean domestication story into a staggering temporal perspective. The ancestors of all modern camelids originated in North America roughly 40 to 50 million years ago. The earliest known camelid, a small, rabbit-sized animal called Protolopus, browsed the forests of what is now Utah and Wyoming. Over millions of years, camelids diversified and grew larger. Some migrated across the Bering Land Bridge into Asia, giving rise to the dromedary and the Bactrian camel. Others moved south through the Isthmus of Panama during the Great American Biotic Interchange, roughly 3 million years ago, and spread across South America. The ancestors of the guanaco and the vicuna adapted to the harsh, oxygen-poor environments of the Andes, developing the physiological traits that make modern camelids so extraordinary. Llamas and alpacas have oval red blood cells, a trait unique among mammals, which allows them to thrive at altitudes where most other animals would suffocate. Their blood carries more hemoglobin per unit volume than almost any other mammal, and their hearts are proportionally larger than those of comparably sized animals. These adaptations were not engineered by the herders of Tulán or the breeders of the Inca. They were inherited from wild ancestors who spent millions of years surviving in the most extreme terrestrial environment on Earth. The domestication of llamas and alpacas was not the creation of something new. It was the harnessing of an ancient lineage that had already conquered the Andes long before the first human ever set foot on a glacier. What does this mean for the llama standing in a field today, or the alpaca whose fleece you see woven into a scarf? It means that the animal in front of you is the product of one of the most complex and least understood domestication stories on Earth. The llama was domesticated not once but twice from 2 different wild guanaco populations in 2 different regions of the Andes. The alpaca is not what we thought it was. It is a hybrid creature, a blend of 2 wild camelid species shaped by thousands of years of human selection, and then nearly destroyed by a colonial catastrophe that erased its original genetic identity. The fine fleece that makes the alpaca valuable today is the last visible echo of a vicuna ancestry that has been almost completely swallowed by llama DNA. The genes inside every modern llama and alpaca carry the memory of that catastrophe, written in the mixed-up code of a lineage that was broken apart and then reassembled from the pieces. Somewhere in the high Andes 6,000 years ago, a guanaco stopped running. We do not know which guanaco. We do not know which night. We do not know which valley or which people first looked at this wild animal and saw not prey, but a partner. But the descendants of that decision are still here, standing in fields and on mountain trails, carrying in their DNA a story that stretches back before the Inca, before the pyramids, before writing itself. What the DNA finally revealed is not just where llamas came from. It is how much of them we have lost, and how much of what remains is a ghost of what once was. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.